Good evening, all of you. In today's PPT, we will be discussing about the plant tissue culture based on the ICAR syllabus for NET, ARS, SMS, and STO. So, we'll be discussing introduction, history, and some of the terminologies used in plant tissue culture. In the earlier video, I have given the detailed syllabus according to the ICR. So in that syllabus, you can see Unit 5, that is biotechnology of root crops. So in order to learn this unit, first of all, we should have a basic idea of the plant tissue culture. For that, we can refer any of the basic textbooks. In this PPT, we will see these topics: introduction, significance, history, and basic principles. So what is plant tissue culture? So basically, plant tissue culture, we will be culturing cells, tissues or organs or even whole plants in an artificial growing medium under aseptic conditions. That is what is done in plant tissue culture. So here you can see the definition. The in vitro culture of plant cells, tissues, organs or whole plants in artificial medium under aseptic conditions. And why is tissue culture possible, plant tissue culture possible? It is because of the two central concepts, that is plasticity and another one is totipotency. So because of this plasticity and totipotency, plant tissue culture is possible. And what does this plasticity and totipotency means? So plasticity means the ability to adapt to changing environment. So when we are taking a cell or tissue and keeping in vitro condition and giving the growing media, they are able to adapt to that changing environment and they are developed to differentiate and grow. So that is plasticity, the ability to adapt to changing environment, that is termed as plasticity. And the next concept is totipotency. Totipotency means the ability of a cell to differentiate and develop into a whole plant when given the suitable conditions. So why this cell is able to differentiate and develop into a whole plant? Because every single cell is having the genetic potential of the parent plant. So this ability of a cell to differentiate and develop into a whole plant, when we are placing them in a growing media, it is termed as totipotency. And this totipotency concept was also mentioned in the cell theory by Sheldon and Sheen in 1837. And this scientist Haberland, that is Gottlieb Haberland, he has done many research works, and from his experiments he found that plant cells are totipotent, and he is considered as the father of plant tissue culture. So the father of plant tissue culture is Gottlieb Haberland. Through his experiments, he concluded that plant cells are totipotent. So here we will see the history, like what are the important landmarks in plant tissue culture. So here I have given the scientist, the year, and what is their main contribution. So first, Hanning in 1904, they have done the embryo culture of selected crucifers. Then Robin Slayback, he has done embryo culture technique in interspecific process of lion species. Then White, he has done successful culture of tomato roots and also stated the importance of vitamins in growing media. And the scientists Snow and Gutheret, they have found the importance of auxin as a growth regulator. Then the successful establishment of continuously growing cultures was reported by Gutheret, Knobcote and White. Use of coconut milk containing a cell division factor was first time reported by the scientist Van Overbeek. Then Skoog and Sui, they found in vitro cell differentiation. Then discovery of kinetin. scientist involved is Miller and co-workers in 1955. So Miller and co-workers in 1955 discovered kinetin, which is a cytokinin. Morel and Martin in 1952, they found the technique of micrografting and found that through micrografting we can obtain virus-free plants. Then Pfizer in 1952, they got the first US patent for in vitro production of secondary plant metabolite. And the plant metabolite they commercially produced was chiconin, and they have done it through cell suspension culture. Chiconin is a plant pigment which is extracted from a plant and it is having medicinal properties. Then Moir in 1953 they have done isolation and culture of single cells from plants. Then Skoog and Miller 1957 they have found the importance or the hormones, how the hormones are controlling the organ formation in tissue culture. Then Reinert and Stewart first report on somatic embryogenesis. So first report on somatic embryogenesis was done by Reinert and Stewart. Then hanging drop method of cell culture was reported by Johns and co-workers in 1960. Then first successful report on in vitro propagation in orchid Cymbidium. So in vitro propagation of Cymbidium orchid was first successfully reported by George Morrell in 1960. Then coming to the other scientist ICAR-ASRB, they have done the isolation of protoplast by enzymatic degradation method. Murashige and Skoog 1962, the development of MS media. Guha and Maheshwari, they have done the production of first haploid plant through anther culture. So Guha is a women scientist from India. From the name you can understand they are Indians. So Guha, that is Shipra Mukherjee Guha. She was from West Bengal, and in the 1916s when the scientific community was dominated by the men, these women scientists have done an important, remarkable contribution to biotechnology through the development of first haploid plant through anther culture, and this was in the crop, if I am correct, it will be the Datura. You can check in the textbooks. Then Power et al. 1970, protoplast fusion. Then the Kabe and co-workers, regeneration of first plant from protoplast. Then Carlson and co-workers in 1972 reported on interspecific hybridization through protoplast fusion. Then Stewart and Reinert first discovered somatic embryo production in vitro. Then this is the general picture of tissue culture. Here you can see the carrot. So we will be taking the cells from the carrot roots and we are placing it in vitro condition, that is inside the glass tube or test tube, and giving the suitable growing media. So there will be cell division in the culture. So from the single cell you can see there will be multiplication through cell division, there will be multiplication, and there will be young plant formed. So from the young plant the adult plant will be developed. So here we can see some of the terminologies used in plant tissue culture. Explant. Explant is a living tissue that we will be transferring from a plant to the artificial medium. So from a plant we can choose any part of the plant, maybe it can be root or leaf or internodal segment or the tip of the plant, maybe flower. So these are all any living tissue which we are taking and transferring to the in vitro condition, that is termed as explant. So here we can see living tissue transferred from a plant to an artificial medium for culturing. It can be any portion of the shoot, leaves, root, flower or cells from a plant. Then in vitro culture. So in vitro, the term in vitro is derived from Latin, or it is a Latin word which means within the glass. So that means we are performing the experiment in a test tube or simply in a glass. So all the type of cultures including animal cells and in vitro fertilization, all for all that we use this term in vitro. The next terminology is micropropagation. So micropropagation means the production of whole plant from an explant using tissue culture. So by using tissue culture, we will be developing a whole plant from an explant. That is scientifically or typically that is the definition for micropropagation. But nowadays, all the terms like tissue culture, in vitro culture and micropropagation are used interchangeably. Usually the method used by commercial tissue culture laboratories is micropropagation. The next terminology is subculture. So uh, in the nutrient media there will be depletion of nutrients after a certain period of time, and also the medium will be dry. So we have to transfer the organs or tissues to the fresh media. That is what is termed as subculturing. So subculturing means after a period of time it becomes necessary due to nutrient depletion and medium drying to transfer organs, tissues, etc. to fresh media. So callus cultures are subcultured every 4 to 6 weeks, and we are able to maintain indefinitely plant cell and tissue cultures through serial subculturing. So we have discussed about the definition of plant tissue culture and also some of the landmarks in plant tissue culture and also some of the important terminologies. Thank you.